Kia ora. No David Chanier toko ingoa. I'm David Chanier from Plant and Food Research, and I'm part of Genomics Aotearoa, and I'll be chairing the session today. So it's a pleasure for me to welcome Associate Professor Mik Black from Otago University. We'll be talking today about the work that is happening around building a long-term local and culturally sensitive data repository for New Zealand Aotearoa. So Mik is leading Genomics Aotearoa project that is about building computing infrastructure and bioinformatics capacity and capability for New Zealand. This is a very successful and a bit of a flagship project for GA. For example, there is a lot of training workshops around the country, led by Mik and coordinated by Ngoni Faya, and this is extremely successful. Most of those workshops are well oversubscribed and they reach to the entire research community from various levels of learnings and many topics being taught. So we'll hear about this repository today. Just a reminder to switch off your microphones. And if you've got any questions, just leave them in the chat box, and we will be addressing those questions at the end of Mik's talk. Just for information, this video has been recorded. And so, yes, please be mindful about your microphones or any comment that you might do. So Mik, yeah, please tell us more about your Genomics Aotearoa Data Repository, and we'll listen to your talk. Thank you.
Kia ora, David. Kia ora tātou, ko whanapaki tamanga, ko maitai te awa, no wakatu, aho, ko Mik Black toko ingoa, kia ora koutou katoa. Welcome, thank you. I'll try and get my screen sharing working. And hopefully you can see that. I'm waiting for a thumbs up from David. All right, that's a good start. Now, Zoom is covering part of my screen where there are words, but we'll muddle on as best we can. So a little bit about me, for those of you who have not met me before. I'm from Nelson originally. Came to Dunedin via Christchurch, where I did my undergraduate work, and then the US, where I was at Purdue University, and then I had a brief period as an academic staff member in the statistics department at Auckland. So my academic training is in statistics from Purdue, and what I do as an academic staff member is I teach, and I teach statistics, bioinformatics, biochemistry, genetics, even some pathology. I also do research. Most of the research I do is in bioinformatics and genomic data analysis, particularly in cancer genomics. So where I'm based at Otago, I'm in the Centre for Translational Cancer Research. As David has said, within Genomics Aotearoa, I lead the Bioinformatics Capability Project, also co-lead the Genome Graphs Project with Dr. Joep de Ligt from ESR, and oversee the genomics training activities delivered by GA. And I'm on the science leadership team in my role as bioinformatics team lead. So I have a fairly deep involvement in GA. In terms of what we're talking about today, we're talking about the Aotearoa Genomic Data Repository, which is a fairly major project within the Bioinformatics Capability Build project. So what I'm going to be going through is I'm going to be going through some background to the project, what our motivation was, what we're trying to achieve, what the goals are for the project. We'll get down into some of the detail, not too far down into the detail, and then we'll actually have a look at where we're at with the repository. I won't do a live run through on the website. What I've got is I've got a bunch of screenshots because I didn't want to be bouncing backwards and forwards between slides and the website. But if you go to the website, you'll be able to see all the things that will be on the slides. We'll finish off talking about where we're going with this in the future. Along the way, there'll be some digressions. You may have noticed in the title of the talk, I had the words treaty compliant. So we'll talk a little bit about what that means, and that will be in the context of indigenous data sovereignty. So before we get started, I'm going to do what statisticians do, which is acknowledge people up front, because we usually forget by the end. So a huge vote of thanks to the GA team who have been involved in this. So we've got Libby Liggins there, Miles Benton, Rudy Browning, myself, as part of the core bioinformatics team within GA. And then from the Rakaora project within GA, we've got Ben Teaika and Ben Curran. And that core group from GA have been involved in a lot of the discussions and the development's gone on as this project has moved forward. The people who've actually been doing work in this space are the NESI team, based in Auckland. So we've got John Hu, Brian Flaherty, and Aaron Perkins. We've all been meeting weekly to discuss next steps for the data repository and talk through where things are going. And so it's been a really huge team effort, and it's been a great thing to be a part of. The other acknowledgements I'd like to make are to the research communities or research community that have been involved. We've run some workshops. We've had really good engagement from within GA from our initial development workshop. And then recently we ran a metadata workshop where we were looking at minimal metadata for collection for the repository. And that involved a broad range of genomics researchers from around New Zealand. So I'm really grateful to everyone who's participated in those ways. All right, so getting into some background, where this all starts from, and I need to move my Zoom window to a place, there we go, you're all gone now, where I can see everything, thanks. So where things start, if we go right back to the beginning of Genomics Aotearoa and the funding agreement with our funder, the Ministry of Business, Innovation and Employment, and that funding agreement, in terms of things that are to be delivered, contains the following text. It says a national genomics data repository, including bespoke processes for Māori management of indigenous data, which is actively populated across all New Zealand genomics research activities. This is a fairly broad range of things. It also does give us a fairly clear indication of what's required. Those are fairly clear terms of what we're trying to achieve. The tricky bit is to figure out exactly how to do that. So that's what a lot of the work has been on, laying the groundwork and then doing the initial implementation of a repository. So just continuing on with our background, there's wording in there about indigenous data. So just in terms of how we're defining that, particularly in the context of New Zealand, a good place to look at that is the Principles of Māori Data Sovereignty, published by Tamana Rarunga. That's available via PDF. There's a link at the bottom of the screen there. So Māori data refers to digital or digitizable information or knowledge that is about or from Māori people, our language, culture, resources, or environments. So that's the definition we're working with. It covers a huge range of potential applications. And in the context of what we're trying to achieve here with a data repository, in particular a genomic data repository, what we've started off by focusing on is genomic data generated from non-human Taonga species. Okay, so now a new definition is needed. So if we're talking about Taonga species, right? What I've done here is I've taken some excellent words that are in the forthcoming Te Noho no Kaitiaki Guidelines for Genomic Research on Taonga Species. This is a publication that has been partly funded by Genomics Aotearoa and will be being released in the very near future. The definitions in there, which are made, are basically taken from the Treaty of Waitangi and moving forward. So Treaty of Waitangi and subsequent documents used to make these definitions. Some of those documents are documents that have come out of the Waitangi Tribunal. So when we're talking about Taonga, it's often used in the context of the meaning of treasured. So Taonga are defined as anything that is of value to Māori. And then furthermore, Taonga species include all indigenous flora and fauna. And then genetic material of Taonga species falls within the purview of kaitiakitanga or guardianship. And so any research in relation to Taonga species and its genetic material is of interest to Māori. So that's kind of the baseline that we're starting from when we're thinking about Māori data or indigenous data and what needs to be stored in the repository. If we're thinking about genomic data, there are a lot of different types of genomic data. So some examples, we might be talking about DNA sequence data, right? So there we're talking about data that could be used to assemble the genome of an organism. When we sequence an organism, we don't necessarily get the entire genome. We will often get tiny little fragments of it, and we have to do some bioinformatic work to build those fragments back up into an entire genome. It might also be information about genetic variation within a population. So there it is looking at the genetics which are different between different individuals in a population. And a really good example of this is DOC's Kakapo sequencing program, so the Department of Conservation, where they have sequenced the genome of all living kakapo. And so there's information about the genome itself, so what the actual structure of the genome is, and also information about the genetic variation that we see within individual kakapo, so between individual kakapo within that population. Another type of genomic data, just as an example, is what we call RNA sequence data. This is more dynamic information about gene activity in specific tissues or conditions. And also another one, again, that's reasonably common is metagenomic data. And this is where we've got DNA sequence data from a collection of organisms. And that could be DNA that's been extracted from an environmental sample, like a soil or a water sample. And we can use that to help identify which organisms are present or absent in that particular environmental sample. So all of these types of information, looking at the relationships between individuals within a population from a particular species, between species and their environments, and can therefore be considered as genomic whakapapa, because we're really thinking about a lot of relationships when we're generating these data. When we're thinking about the storage of data, we have some guiding principles. Some of those principles are summarized by the FAIR data principles. And so this was published back in 2016. And it was the idea that scientific data should be findable, accessible, interoperable, and reusable. So we should actually be able to discover that data. We should have the means to discover it. It should be put somewhere it can be found. We should have the means to access it. So we should be able to obtain access to that data through some mechanism. It should be usable. We should be able to use it with our software. We should be able to integrate it with other data sets, and it should be reusable, where it can be repurposed for other things. So other questions can be asked of that data. Now, I just want to make a point here that the FAIR data principles are often misrepresented as, or misinterpreted as advocating for what we call open data. Now, open data are universally publicly accessible. So anyone could download a data set from the internet. That would be a definition of open data. Whereas FAIR is only saying the data need to be accessible. And that access could be by an application mechanism. So you have to apply for access. So that would still fit within FAIR data principles. However, in scientific research, making genomic data open is actually often the default position. So often the default, and it's required by a lot of academic journals. So when researchers submit their research for publication, they are often required by that journal to make their genomic data publicly accessible. So I've written traditional here, inverted commas. I possibly should have written typical handling of genomic data. So what typically happens in scientific research, data and knowledge sharing are really fundamental concepts that underpin what I've written here today is the Western scientific process. There I'm meaning the typical research that is done in academic or research institutions around the world. Now, for non-human genomic data, and also a lot of human genomic data, there is the expectation that researchers will make their data completely openly available. There are some good reasons for this. It makes data sharing a lot easier. So it means that researchers can get access to data very easily. It does a lot to help ensure scientific integrity. So it ensures the integrity of both the data, so we can easily check the quality of a data set, and it also helps to ensure the integrity of the analysis methods that were used and the analysis that was performed. So having data as open goes a long way for science to make sure that good science is being done. It also improves value through data reuse. So it allows other researchers to download data and use it for other purposes. And in my own research, I have made huge use of publicly accessible data, particularly in the cancer space, where we have integrated data sets from all over the world and use those to help with our own research. There are a lot of repositories that handle this around the world. And the way I've described it, hopefully makes it sound like wonderful goals to have in terms of this openness and the sharing. Now, there are problems. And problems were sort of pointed out a few years after really the open science and open data movement became very prominent. And these were problems that open data were generating for Indigenous communities. And so what was developed out of those concerns by the Global Indigenous Data Alliance were the CARE principles. And so the driver there was that the current movement towards open data and open science does not fully engage with Indigenous people's rights and interests. So the CARE principles talk about collective benefit, where the mechanisms used to host and share data also enable indigenous peoples to derive benefit from that data. So there's some mechanism of benefit sharing built into the process that indigenous peoples have the right to control the data that relates to them. And that those who are working with data from indigenous communities have a responsibility to ensure that those benefits are actually being realized and that the research is being done in an ethical way. So this was almost a little bit of a pushback against that very strong drive for openness that was coming from a large chunk of the scientific community. Within New Zealand, there's been some excellent work done in this space where Te Mana Rārunga have reframed those care principles into really a Māori values context, putting those components of care across the various components listed here. So rangatiratanga, whakapapa, whanaakitanga, kotahitanga, manaakitanga, and kaitiakitanga. Just delving into a couple of those, they really do fit quite nicely into the care principles and reflect what's going on there. So particularly, and thinking back to that slide earlier about the treaty, thinking about the authority to have control over data about Taonga species, and also being able to have kaitiakitanga or guardianship over the Taonga species, and being able to derive benefit from the use of data from those Taonga species. So when we take all those principles, combine them with the FAIR and the CARE guidelines, those provide a really strong underpinning for what we've tried to do with the Aotearoa Genomic Data Repository. Now, an important point to make here, and I'm going to probably misquote Ben, but I'll do it anyway. So once our data leave Aotearoa, they lose any and all protections provided by the Treaty of Waitangi. So this is a really important point. It's one I hadn't completely appreciated before we started talking about this, but it reinforces that not only do we need to acknowledge those FAIR and CARE principles, but also we need to think about housing genomic data from Taonga species within a New Zealand-based platform. Because once data leaves New Zealand, we then have no jurisdiction over that data. We don't have any control over what happens to it. It also reinforces the importance of involving those who have kaitiakitanga over the data in decisions about access to the data. So with that as our framework. No, and I've thrown a slide on some extra reading. There's some great work that's been done in that space. Publication involving Maui Hudson there around operationalizing, I guess, putting those care and FAIR principles into practice for Indigenous peoples, and then article in Nature Reviews Genetics by a whole range of New Zealanders. There's some of the GA team around use of genomic data for Indigenous peoples. Right, so with my previous slide as the backdrop, what we're looking to do with the Genomic Data Repository is establish a New Zealand-based resource so that we can store, manage, and share genomic data from Taonga species. We need a system that lets our researchers and our kaitiaki fulfill their responsibilities relating to genomic data sets. Our researchers have responsibilities in terms of data integrity and data sharing and data accessibility. Our kaitiaki have those guardianship responsibilities. And we want a system that allows both of those sets of responsibilities to be fulfilled. What that means is we've really got two components to the project as we go forward. We've got a technical component where we're thinking very deeply about, how do we implement and deploy a compute infrastructure that underpins the repository and accomplishes these goals. And then we've also got a cultural component where we're thinking about how do we develop protocols that cover the data submission and also cover accessing the data, and what information do we need to collect when data are being submitted, when access requests are being put in, that will enable those requests to be assessed properly. So who you're going to call? Probably not Ghostbusters. Instead, we're going to call Nessie. So Nessie is our New Zealand eScience infrastructure. Most of you are probably aware of that. We already have a very close relationship with Nessie. Nessie are our partner for providing compute and storage resources for all of the Genomics Aotearoa research projects rather. We also have a partnership, David mentioned earlier about the training we run, that is done in partnership with Nessie. And we have a really close partnership on delivering genomic skills training nationally to our researchers. We also are involved in community building, particularly around our national e-research community. NESI is a big team. You can see photos all down the screen there, and they have a really, really broad range of expertise. And so that was one of the other reasons that we want to work with NESI on this, because we have access to a bunch of fantastic people who have an incredible skill set. Particularly relevant for the data repository work is their expertise around storing research data, large-scale compute, and in particular security, particularly around data security. Who else do we call? Ben's going to be embarrassed because he's sitting just outside my office there somewhere, but we called Ben. So at the time we did a lot of this work, Ben was the Vision Maturanga coordinator for Genomics Aotearoa. So Ben has now gone on to be the project manager for the Rakiora project within GA. But in his role as VM coordinator, Ben did a huge amount of work developing a really extensive set of protocols for data submission. So figuring out what questions we need to ask when data are being submitted to capture that key metadata around the consultation process that has been gone through by researchers before they generate genomic data from Taonga species, and also the kaitiaki details associated with that particular genomic data set. Also protocols around data access requests. So what information do we need to capture when people are asking for permission to use a data set? Some of that, we'll get to more of that later, but some of that is what is the research being proposed? What benefit is there to Māori? What consultation process have they gone through? And that is all information that is then used by the kaitiaki for that data set to make a decision about whether or not to grant data access. So what have we ended up with? So the outcome, very quickly in the one slide, by combining those technical efforts with the cultural work done by Ben, we've ended up with version 1 of our data repository. And so from a technical perspective, and I'll go through some of these details in a little bit more detail in the next couple of slides, from a technical perspective, we've ended up with the Gen 3 Genomic Data Repository platform. So there's an open source platform for storing genomic data. Linked in with the Globus system for high-speed data transfer to allow us to move data very rapidly in and out of the repository. Some of the data sets that we're dealing with are reasonably large, and so they benefit from having a specialized data transfer tool there. And this has been deployed onshore using NESI compute and storage resources. So this is physically located within New Zealand. So fulfilling that requirement that we had about having to have things located onshore here. From a cultural perspective, we've taken those fair and care principles. We've used Te Mata Ira, the guidelines for genomic research with Māori, as the ethical framework that underpins a lot of the work that Ben did. And then Ben's created that kaitiaki-centric data access and submission protocol component there, which then controls what information we collect and how access is granted. So just thinking about Gen 3, we're talking about Gen 3. Gen 3 is an open source project. It was developed at the University of Chicago in collaboration with the National Cancer Institute in the US. It is used in some fairly major implementations worldwide. It's kind of interesting to me because one of those implementations is the Genomic Data Commons at the National Cancer Institute, and that hosts data from probably 15 to 20,000 tumors, huge amounts of genomic data, and it's one that I use all the time for my own cancer research. And so it was interesting that when NESI went out and they had the brief of basically figuring out what existing genomic data platform would be best for us to provide. And so it was interesting to me when they came back and said, hey, we've found this thing called Gen 3. It looks really good. They use it at the NCI. I said, I know, I use it all the time, which was great. So I didn't force the picking of that one, but it just happened that that was the one we came up with. And it's one that I was familiar with through the research I've done, and it works really well. So another bonus of choosing an open source platform is that we get a lot of valuable work that's been done, or access to valuable work that's been done, and setting things up by University of Chicago and the National Cancer Institute. So all of the work that those teams did and getting to the point where they are today with this platform, we're able to benefit from that. And actually the NESI team have had some fantastic interaction with the Gen 3 development team. When we've hit a few snags over here with implementation, they've been very responsive and helping out with fixing things up and responding to our inquiries. So open source, freely available source code, very active community, and very great support coming from there. Another important thing is support for what's called data federation. The example here says we can point to data on the NCBI. So this is really another data warehouse in the US where a lot of genomic information is stored. If we wanted to, we would be able to have content within the data repository that would point through to the NCBI as if it were in our own repository without it actually being there. A more important point from the perspective of indigenous data sovereignty is that we could also have a data set that was hosted elsewhere. For example, it could be physically hosted on compute resources owned by an iwi that we could point to. So it would appear as if we had that information in the repository, but it would never actually physically leave the control of the group that had kaitiakitanga over that data set. So that's another feature that we're looking at as part of Gen 3 that is potentially advantageous there. In terms of data transfer, I mentioned this before, Globus is the system that's actually been deployed for a number of years now in New Zealand, and we've been using it to transfer genomic data around the country and around the world. It's a framework for high-speed research data transfer. I took a snapshot yesterday of how much they've processed. I don't know if you can see my mouse on here, but this is megabytes, gigabytes, whatever comes after that, terabytes, petabytes. That's a lot of bytes. So this tool is used to transfer huge amounts of data around the world. You'll see we've actually got a Kiwi on here, and that is thanks to NESI being a national cyber infrastructure provider who is signed up as a member of the Globus Alliance. So within New Zealand, NESI and our Research Education Advanced Network New Zealand have been providing national support for Globus for a number of years. And that's what we're using for data transfer in and out of the repository. So it is a fairly user-friendly, somewhat user-friendly mechanism for enabling very fast data transfer, either to the repository from an institution or in the other direction. Oh, that snuck up on us. We're in. Okay, so let's have a look at this thing. So what have we actually put in place in terms of a data repository? The link is down here in the bottom right of the screen. And this is what you will hit if you go to that link. This is the landing page. And straight away, you get some information about what's in there. There's not a huge amount of data in there at the moment. What we're looking at is we're looking at 9 projects, so across a range of organisms. We break those down into sub-projects. We're probably going to rename experiments, but you can think of an experiment as a subcomponent of a project, and then individual files that are in there. The color coding down here is just showing what percentage of each of these different objects is made up by, in this case, the Kakapo project. So it's one of the projects. It's a bunch of sub-projects in there. There might actually only be one sub-project in there, but it's actually a huge percentage of the files that we have in the data repository at the moment, because it's a fairly big project. From there, if you're interested in having a look at what projects there are, across the top here, there's a bunch of tabs. You can get to the projects either by going up into the top at the middle there, clicking on projects, or the big green button down here. I don't have to push it because I've already taken a screenshot. Within that projects interface, you get a listing of all the projects that are in there. I've just taken the top 3. There'll be another 6 underneath. As we get more projects coming in, we are probably going to have to alter the way in which this is displayed. So we'll have to add a search feature so that you're able to search through for projects, because at the moment it's just simply scroll down to see what projects are in there. And then if you click on one of these show details type links, you will, not that one. This one is looking at the Manuka project from Plant and Food Research. So when you click on the details link, you are taken to a screen which has some more details about the project. That is the details that you get are dependent on the information that we were provided with when the data was submitted. One of the things you'll see here, I can say this because David's online, it's a little bit sparse. It would be lovely if we had a little bit more information about this particular project, maybe a nice glossy photo of Manuka. But it provides some basic information about what is the project, and in this case, the data set contains gene predictions and a genome assembly for the Crimson Glory variety of Manuka. On there is some information about who to contact about the project. So Dan Jones is listed on there with his contact details. Now, that's not how you get access for the project. So Dan is the researcher listed as the contact for this project, but he's not the kaitiaki of the data set. And so if you are going to try to get access, there's a process to go through, and we'll talk about that a little bit later on. As you might expect, it's by clicking this big green button here, and that will begin the process of applying for access to this particular data set. But we'll have a little bit of a look at some of the other features first, and then we'll have a look at the data access process there. Right, so if you move across to the exploration tab at the top, we end up in an interface which, to anyone who's ever been to the National Cancer Institute in the US, looking at their genomic data repository or their genomic data archive, this will look pretty familiar in terms of the setup. So this is where you start getting into the nitty-gritty of what's actually stored in this repository and thinking about what sort of files you might like. So when we come in here, we get a listing. If we click on the experiments tab here, and we can start doing some filtering by the metadata that's associated with these samples that have been submitted. You can see up here, we've got geographic location, we've got some latitude and longitude stuff down here, development stage if we're thinking about development of organisms, sex, etc. So there's a whole host of metadata that we can filter through. Just looking at the geographic location, and the most obvious thing is that we don't have a lot. Right, so for 13 of the experiments in there, we don't have any information about geography. We've got some fairly coarse information about some, South Island or North Island, one from Palmerston North, thank you Plant and Food, and then one from Inner Tasman Bay. It would be really nice to have more information about where these samples have come from. So this is where we need to do work in terms of encouraging researchers who are making submissions and going back to them and just trying to tease out some more detailed information about that. The latitude and longitude one, as we're talking about, that would be really cool to have that sort of information available. One caveat to that is that that information could actually be quite sensitive. So if we're thinking about exact coordinates of where samples were taken from, particularly when we're thinking about Taonga species, that itself could be very sensitive information, and we may not want to make that available, or we may not be allowed to make that available. Obviously, those coordinates can be rounded to a much more coarse value, so we can see the general area or maybe province in which a sample was collected. So really just showing here that we have the facility to incorporate a lot of these different types of metadata, but also that we don't have to make all of that metadata available. In terms of the metadata, I know I've got one more slide before that. So clicking over into the Files tab, again, it just gets down into more of the nitty-gritty about what's actually being stored in the database. So here we can see file counts per project. Again, you can see that Kakapo project's got the most files in there. For the genomics aficionados, you can see what types of files we're dealing with. So we've got basically raw sequence files coming off the sequencer as FastQ. We've got FastA for like genome assemblies, and we've got BAM files for aligned data. So we just get a breakdown of the different types of files that are in there. So these are all genomic data file formats, and then also a breakdown of the proportions of files from each of those projects in there. And then we get a listing of individual files coming up one after another. For those who are really interested in the structure of what's going on and what's being captured, you actually get the ability to delve into what's called the metadata dictionary. So this is how all of the information that's being collected, not the genomic information, but all of the additional information that comes along with it, how it's collected and organized and how it relates to each other. And so at the top here, we have what's called the program. And the program is basically the top level of the repository. So it's everything that is in the repository. And in this case, our program is called Taonga because all of the things within our program are in this Taonga species database. Within our program, we have different projects. And we saw some of those before. So the Kakapo dataset was one particular project. We can then have different sub-projects within that project. And I mentioned before, those are currently called experiments. We're probably going to move away from that because in most cases, what's being collected as a sub-project isn't really an experiment. So we'll be looking at renaming that to make it a little bit more intuitive. And then you delve down from there. So as you go down, there's more and more detailed information, and you can see how it is organized hierarchically here. If you click on something here, if I click on Indigenous governance over here, what will happen is it will pop open a little subtree here, and it will tell me something about this particular node. So it says within Indigenous governance, there are 3 required properties and there are 7 optional properties. If I click to open those, I get a screen popping open. I note that we need to add some descriptions for some of the things in here. And you see that 3 of those are required fields. So projects, what project is this thing called, who's the submitter, and what type of data set is it. Definitely we can handle some descriptions there. And then we see a bunch of non-required fields, and you might actually get quite concerned because we've got a lot of information about kaitiakitanga, which we're saying is not required. A word of explanation. So for some of the data sets that have come into the repository, we don't have that kaitiaki information available. If we made that required information, we wouldn't actually be able to get the data set into the repository. So we've relaxed that so that we can bring the data set in, but we've also noted that there is no kaitiaki information if there isn't, or if there's kaitiaki information that's recorded in there. So that's just an explanation of why we've got non-required there. What we'll be working towards is that when data sets are submitted, that it is a requirement to provide that kaitiaki information as part of the evidence of consultation process that you've been through. I'm seeing a few things in the chat window popping in the chat thing popping up, but I'm not looking at the window. So if anyone desperately has a question or needs clarification of something, do feel free to just pop your mic on and ask a question. Right, something to note. So I've said I'll talk about the data access request process in a moment. But something to note, particularly for the Kakapo project, but it illustrates an important point. If you click on the Apply for Access button in the Kakapo project, what you'll actually get is redirected back to the Department of Conservation website. And that's because for this particular data set, the Department of Conservation has their own data access process that they work through. And so there you're directed back here and you'll work through the doc process in order to request access for the data set. Now, if you get through that process and are granted access to the data set, what will happen is doc will get in touch with Nessie, provide them with the details of whoever it is that they have provided access to, and Nessie will then arrange for access to that within the repository. They'll set up the Globus links and make it available. So what this illustrates is that you don't actually have to use the access and approval method that's been developed by GA. If you have got an established access and approval method available, we can work through that one through an external pathway as well. So just an illustration of one, what happens if you request access to the Kakapo data, and two, illustrating that there are other pathways that can be used for generating access requests. Just final thing to highlight under support, in the tabs, is the support tab. If you click on there, you actually get taken to, you're taken out, sort of out of the data repository to a support interface, where there's actually a huge amount of support, huge amount of information about how to navigate around the repository, how to get access to data. If we click on the next one, and how to set up your Globus endpoint to enable Globus access in and out of the database if you're uploading data or if you're accessing data. I will say some of this is slightly out of date at the moment because we've done a lot of work over the past couple of weeks on the repository. So we are going to have to go back through and make a few updates to our support documentation. But just be aware that there is quite a lot of documentation there around how to use the repository and how to get information out of it. So if we're thinking about data submission, back on the front page, there are a couple of buttons down in the bottom left corner. One was projects, the other was submit data. So if you click on the submit data button, you will be taken into a form. At the moment, that's a Google form. We are looking at whether there are other options to integrate in with the repository. But the sort of information that you have to work through are the basic sort of thing, who are you? What are your contact details? What species are you working on? Where did it come from? GPS coordinates. Now, GPS coordinates are an interesting one. I mentioned before that those might be sensitive. The GPS coordinates aren't necessarily for inclusion as metadata or inclusion as data as part of the project. They're actually to aid with the assessment of who are the iwi, what iwi may have kaitiakitanga over the species you're working on based on the geographic location from which it was sampled. So this is information that can be considered when thinking about kaitiakitanga, because you know exactly where those samples have come from. So this is one of the things that Ben was very keen to have included as information, because it provides, it's so useful in terms of who do we need to talk to when we're thinking about who has responsibility for these particular samples. In terms of consultation and kaitiaki, there's information requested about the consent and consultation process that you've been through as a researcher to generate these data, and also information requested about kaitiakitanga for the data set. And there's fairly typical information about research institution, what the contacts there are, and then down into the more nitty-gritty about what are the files, how big are they, what are you trying to upload? So it's about 4 pages of information that you have to go through in terms of making the submission. And then there's additional forms to fill out in terms of the metadata about the actual data files that you work through with the NESI team. If you are requesting access, same thing. You click on the button, apply for access, you end up in a form, you select the data set that you are interested in. I think that's automatically tagged. If you're at a data set and you click apply for access, it automatically knows which data set you want. And then it sends you to a similar form. So looking for research name and contact details, whether or not the data originator or a Māori authority holder has already consented for you to have access to that data. Information about any Māori consultation that you have undertaken as part of the research that you are doing or proposing to do with the data set you want access to. So this is not consultation that has gone on to do with the original data set being generated. This is consultation in the context of the research you want to do by getting access to these data that are in the repository. So information on that, information about how you'll handle risk to Māori relationships, values, rights, and interests. Whether or not any recombinant DNA techniques are being proposed for use in the research that you're proposing, information about what are the benefits to Māori from the research you're proposing, information about how ownership and IP sharing will be handled, whether there are any aspects of the research you're proposing that are culturally or commercially sensitive, how well your research aligns with the Te Mate Ira guidelines, which presupposes that the researchers requesting access are familiar with the Te Mate Ira guidelines, and then some more specific information about the research institutions they're coming from, what the project actually involves, what the sources of funding are, who's on the team, that sort of thing, and a whole bunch more stuff. So I may have gotten towards the end slightly. I know I'm not doing too bad for time. So the broad brush summary is that version 1.0 of the data repository is up and running. It is still called the beta at the moment. The link is there. We will be doing an upgrade to the URL. So we will be moving to agdr.org.nz in the nearish future. There are a few technicalities we're working through within the NESI system about how the virtual environment that the repository runs on is actually hosted. Once that's all ironed out, then we'll upgrade to the new URL. Currently, researchers can deposit data at the repository. They can browse through the repository. They can put in access requests to the data sets. We're storing information about kaitiakitanga and a whole bunch of other stuff as project metadata within the repository. And importantly, those kaitiaki are involved in decisions about getting access to the data. What's coming? So what are we working on? Currently, we're working on some of the technical aspects of searching through the metadata for finding things that you're interested in. On the next slide, I'll talk a little bit about integration of traditional knowledge and biocultural labels into the repository. We're working on keeping the documentation up to date because we know at the moment it's slightly out of date. Also working to get some additional project information from our submitters. So we've got some richer information about each of the projects. I said we had that workshop on minimal metadata requirements. So we're working to finalize those requirements so that we can make more recommendations for people submitting. Also establishing an external Māori advisory group who will be responsible for really helping guide the ongoing development of this. And in the future, we're looking at actually being able to visualize some of that geographic information that's in there. One of the really nice features about Gen 3 is that it doesn't just store data, it actually allows applications to access into the repository and work with the data that's in there. So what that means is hopefully in the future, if there are data sets where there is, say, summary data which can be publicly accessible, or whole data sets that can be publicly accessible, we can have applications that sit on top of the repository where you're actually able to go and do some analysis. Or if you've gained access to one of the data sets, that you'll actually be able to go log in, get access to the data, and then interact with it through a web-based interface. And we're also working on getting more data in there. We have got a number of data sets queued up to go in from groups around New Zealand at the moment, and we'll just basically be working our way through that submission process. Just a couple of quick words about the labeling. We are working to make this part of the repository. Localcontext.org provides a whole lot of information about how these labels can help Indigenous communities with managing their intellectual and cultural property. Broken down into two categories, so traditional knowledge labels and biocultural labels. Probably for us, most of the utility there would be in the biocultural label space, but this mechanism allows Indigenous communities basically to create these labels which specify things like attribution rights, provenance, who's allowed to use the data, what the consultation process that was gone through to generate a data set, and then they can assign those labels to a data set. We can incorporate those labels into the data repository, and so when you're browsing through the data, you can click on the label, it'll link back through to the labels at local context, and you'll be able to understand what the basically anything that's been attached to that label by that Indigenous community, you'll have access to that information. So we're working to get that integrated into the repository now. If you're interested in having a bit of a read about some of the work we've done in the data repository and also just the use of metadata in general, there are some nice case studies, one from Dr. Kim Hanley at Auckland, one from Dr. Libby Liggins at Massey, on those things, just to help give, I guess, a deeper understanding of what's going on with some of the repository work. And then finally, I would like to thank again all of the team members that have been involved, but in particular the NESI team. June, Brian and Iron have worked incredibly hard on this and are really passionate about establishing an effective and functional repository, as we all are. I would really like to thank Ben Tiaka for all the amazing work he did in developing those submission and access protocols, and also for everything I've been involved in, not just the GA stuff, all my research and teaching work as well. I've had some fantastic advice in the Tā'awa Māori space from people like Ben, Phil Wilcox, Alana Alexander, Maui Hudson, have all been very, very generous with their time. So thank you for that. Finally, I would like to thank the Ministry of Business, Innovation and Employment as our funder, and NESI and our GA partner institutions, and all of you for your attention today. So thank you.